So a little bit of an overview about Revivicor now that you've gotten through my technical issues. We started out as the U.S. division of a Scottish-based company called PPL Therapeutics, where at that time our focus was on cattle as a bioreactor for proteins in milk. But in 2003, we spun out as a standalone company, venture capital funded company. And usually what I'm talking to you about is xenotransplantation. And our focus before we were acquired by United Therapeutics was on medical devices, organs, and islets. But then when we were acquired by United Therapeutics, now I didn't have to worry about raising money every day and could actually focus on the science. And at that time, our focus became on food, which I'll talk about as our agricultural product for the GalSafe pig. What I won't be talking about today is our efforts in xenotransplantation of kidneys, hearts, and lungs. But as you can see, this all started with the Dolly technology, and then we cloned Mr. Jefferson, which was our first cloned cow, and then the world's first cloned pigs. I want to talk about the alpha-gal story, and Tom brought this up earlier. This is an issue for people primarily living in the southeast part of the United States, but for those internationally, there is a tick vector in every continent except for Antarctica. And so in the United States, that tick that is the carrier is primarily the Lone Star tick, although there's evidence now that the black-legged tick and chiggers can also transmit this alpha-gal allergy. The tick vector, biting, let's say, a deer or a mouse, and then taking a meal on the human as a tick bite and injecting some reconstituted protein from that other mammal. And that essentially is injecting an alpha-gal into the patient, which is the alpha-gal syndrome person. So it's actually an unusual disease. It can take anywhere from 1 to 3 months before your allergy antibodies, IgE, become elevated to a point where now, in addition to your tick bite reaction there, you can become reactive to a whole variety of mammalian-based products. Mainly today, I'm going to be talking about the food products coming from the pig as pork. But then if you have this alpha-gal allergy, you can be highly allergic to blood products, whether there be heparin or pancreatic enzymes or any number of gel foam and things, pharmaceutical drugs like vaccines, gelatin-coated capsules, as well as tissue products. We all know that heart valves for many decades have been coming from porcine or bovine sources, or sheets of tissue coming from small intestine, submucosa of pigs, bone. All these things can be issues for these patients that have the alpha-gal allergy. The mitigation in this case is a benefit to the consumer. In this case, a patient that has this alpha-gal allergy can have a mitigation because of animal biotechnology, gene editing. And actually, we did this back in 2001, 2003, that timeline, where we were using just standard homologous recombination to make a targeted insertion of a PPL 657 vector. So the IgA, or the intended genetic alteration, is inactivating the GGTA1 gene, resulting in animals that are completely alpha-gal negative, so they don't have this alpha-gal sugar. And because they don't have the alpha-gal sugar, they actually have anti-gal antibodies, and so they can actually be used as a model for other applications in testing reactions to alpha-gal. But as you can see in the lower left-hand corner, you can't tell any different between an animal that is gal null, that's alpha-gal deficient, versus an unmodified pig that has normal levels of alpha-gal. Over the last 20 years, we produced more than 1,000 of these pigs. I'm not going to call them knockout pigs based on what Allison was saying, but these pigs have been produced over 15 generations. We've been able to show that they have normal growth, normal fertility, normal health in all aspects, things that are very important to the FDA during your approval process. We're at this point only maintaining these pigs in the United States in 2 herds, one in Iowa and one here in Blacksburg, Virginia, where I am. And we've actually now moved ahead with producing this meat in a USDA licensed abattoir, which is going to be important for these patients with alpha-gal syndrome, otherwise known as the red meat allergy. But once you have this meat in a USDA approved abattoir, now you can think about taking all the active pharmaceutical ingredients that come out of, an unmodified pig abattoir, things like heparin, collagen, heart valves, orthopedic tissues. All of these things can be coming out of the abattoir in addition to the meat products. And so there's a huge benefit for these alpha-gal syndrome patients to have these other medicines available to them that are a byproduct of the meat production. Now, our parent company is also looking at ways to produce organs beyond xenotransplantation. So you can also be looking at producing scaffolds, decellularized scaffolds, in this case, a lung scaffold, as in the right-hand corner, and be repopulating those with human cells and using that as an alternative to have an unlimited supply of donor organs. This is what I'm going to be talking about today, which are the agricultural products, or primarily food from the GalSafe pig. And as others have talked about, there's a very directed method that the FDA goes through, and we applied for our new animal drug application starting back in 2008. And there are multiple steps which I'll be going through, as you can see in the binders down at the bottom of the slide. But we got investigational food use authorization granted by the FDA for the GalSafe pig in August of 2019. It was fully approved for both food and therapeutic use on December 14th, 2020, and that was primarily our GalSafe animal operation in Iowa. And then our animal operation in Virginia was approved by the FDA in May of 2023. So kind of not spending too much time on this, but section 1 of the new animal drug application is the product definition. In this case, the PPL 657 recombination rDNA construct that's used for the alpha-gal inactivation. And the ultimate product is a product that has undetectable levels of endogenous galactose alpha-1,3 galactose sugar residues. Next on is the molecular characterization of the construct, which in this case would be that PPL 657 construct. You want to show that you have the intended design of the construct, where various DNA technologies looking at fragment sizes, whether that's through restriction digest or PCR, and then ultimately DNA sequencing of the entire construct. As far as the next phase of the new animal drug application, section 3, which is molecular characterization of the animal, is in showing that the intended genetic modification is as achieved both ex vivo, looking at cells, as well as in vivo in the animal itself, and then proving that to the FDA through all types of genomic application. So as far as ex vivo, we're transfecting that PPL 657 construct into cells. We're actually then selecting the cells that have that intended genetic design, and then using various DNA technologies, screening by PCR, long-range PCR, and Southern blot to show that particular construct and the inactivation of the alpha-gal gene is as intended. So we have to define not only founder animals, but also lineage progenitors. So the founder animals, in this case, were made by somatic cell nuclear transfer or cloning. And then those lineage progenitors are verified by long-range PCR, Southern blot. And then we want to make sure that there was no off-target analysis, which others have talked about as well, and that the targeted insertion is as designed. The phenotypic characterization of the NADA is section 4. So we've looked at things as far as live weight of the pigs compared to normal unmodified pigs, and they're in the normal range. We've looked at the health characteristics, whether it's a CBC. In this case, you can see that the erythrocyte characteristics are the same for GalSafe pigs as they are for an unmodified pig. And also just looking at litter sizes, and again, the GalSafe pigs from 2 different generations are right in the norm for the industry norms for litter sizes. Section 5 of the NADA is a durability assessment and plan. So you want to demonstrate that not only is the construct durable as far as its genotype and consistent and predictable, that there's no alterations of the DNA construct over multiple generations. And then also your production plans, how you're going to be breeding these animals and showing that you've got phenotypic and genotypic identity of, in this case, the GalSafe modification. And of course, any of those through breeding that didn't meet those identities would be considered out of spec animals, which I can say we've never achieved in our lines over 15 generations. Section 6 is the food feed safety assessment. So this is a concurrent review between FDA and USDA, which was very beneficial. It's been a very much of a synergistic discussion and approval process with both FDA and USDA. Of course, there's a risk assessment associated with the food feed safety, looking at the vector itself, the targeted insertion, making sure there's no off-target effects, showing that the alpha-gal is completely null in these. We've actually done a compositional analysis of the food product. We've also looked at taste and texture, and that basically this food production will adhere to USDA standards. As far as mitigation of exposure, it's really talking about making sure these genetically modified animals don't escape, and that you have a method, a regulated method to identify the animals if that were to occur. So it's the idea of triple fencing, where you've got a pen inside a building, and then the outside of that building, it has a fence around it, and showing that those barriers exist not only for housing the animals, but transport, slaughter, and moving the actual product. The other aspect of Section 6 is environmental assessment. This is a public document. So as I said, the FDA approved our first IGA for both human food and therapeutic use. And this is a document that you can go online and find. It's based on facility. So our operation in Iowa is actually where the first environmental assessment and our first NADA approval is based on. Secondarily, when we wanted to set up the GalSafe herd and as a production and FDA regulated site here in Virginia, we had to have the environmental assessment be specific to the Virginia sites. So you're looking at, probability of the pig escaping, the ability to it to spread to the wild population, which of course that's not going to happen unless the animal escapes. And again, coming back to the containment of these animals. Section 7, which is the final part of the new animal drug application under FDA, is the claim validation that this is actually an alpha-gal null animal. And so we not only have to show that the animal is alpha-gal null, whether it's by flow cytometry or immunohistochemistry, and this is also by both genotype and phenotype, show that it's persistent, it's durable, by gender, male, female, by generation, now over 15 generations, and as the animal ages, and have repeated measures of specific animals or sentinel animals in the population. So as I said, this was a simultaneous review by both FDA and USDA, and in 2021 we got approval by USDA of our label. And importantly, you have the GalSafe TM with this secondary label that you see on the right with the AG with the line through it, is important because this is an actual claim. And in this case, the claim is that pork does not contain detectable levels of alpha-gal, which causes allergic responses in certain individuals. And I think this is very important because when you look at something that's gluten-free or vegetarian, in our view, our vision is that you're going to open up your menu in the next couple of years. You're going to have your vegetarian option, you're going to have your gluten-free option, and it's going to be very clear that the option on your menu, if it's AG-free, is GalSafe and does not contain detectable levels of alpha-gal. So we started distributing this food, very low-tech operation initially. As you can see here, this is a compassionate use distribution. So initially, we just are giving the meat away to alpha-gal syndrome patients while we were waiting for a commercial partner for growing the animals and distributing the product. So we're essentially going through the process of storing the meat as it comes back from our USDA-approved abattoir, taking that frozen meat, boxing it up, and sending about 10 pounds to alpha-gal syndrome patients. And where we're getting requests for this pork, is essentially where the endemic area of the Lone Star tick is. There are some folks out all the way out west in California, in the Midwest, in the far west that are experiencing this allergy. But we've been distributing this GalSafe pork since late 2021. We've distributed to more than 2,000 alpha-gal syndrome patients without a single reaction. And just recently, we signed a deal with a Midwest partner for growing the GalSafe herd. And as a result of that, we expect our first commercial sales near the end of this year. But as you probably can't read it on the right, but as we've been distributing this product to these AGS patients, in this case, the genetic modification is benefiting the consumer, not the producer. And I think that's really important because these folks, some of them haven't had, a BLT or any kind of mammalian protein for 2 years, 4 years, 8 years, 10 years since they were diagnosed. And when they get that package and they get their bacon or their ham or their pork chop or their ribs, the amazing opportunity to taste bacon again after 8 years, for example, is just overwhelming to these folks. And we get videos, we get songs, we get all types of things from these patients about how happy they are to finally be able to eat pork products. And so that's really makes this worth it for us as we go through this. So I'll just stop here. I don't want to go too far over, and we can take some questions, I think, at the end. Thank you.